Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com and Indigo Biosciences webinar titled Advanced Water Quality Monitoring, How to Easily Integrate Advanced Bioanalytical Techniques for Comprehensive Insights. I'm Liam Sanyo for the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Jack Vanden Heuvel, professor in veterinary and biomedical sciences at Penn State University, who will discuss how integrating cell-based bioassays and effect-based trigger values enhances water quality assessment beyond traditional methods. And with that, I'd like to welcome Dr. Jack Vanden Heuvel. Jack, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you very much, and thanks for joining us for this talk on a new approach to looking at water quality, and that's using some bioanalytical techniques, including cell-based bioassays. And by the end of this talk, I hope that we have some insights on how we can use mechanistic information for making determinations about the quality of different types of water. So here's the outline of what we're going to cover today. We're going to start with just the basic introduction. What is the problem that we're facing? How do we approach this currently? And then we'll start talking about some important mechanisms that are associated with chemicals present in water. And then we'll go through the process of how you can use this molecular biology information to make decisions about water quality, including using chemical analysis, bioassay analysis, and then we'll finish up with looking at how you can use the bioassays with certain trigger values in order to trigger decisions. So let's look at what our problem is and how we are approaching it. So first of all, when we're talking about water quality, this can really mean different things to different people, depending on what you do, where you live, whether you're a stakeholder or whether you're a producer of some of these chemicals or you're in charge of monitoring safety for a particular environment. We also have to think about how this water is being used. Is this drinking water we're evaluating? Is this surface water, groundwater? Are we talking about water that needs to be recycled and reused for either potable water or for agricultural use, for livestock or for irrigation on fields? Another thing that we would like to do is also protect not only human life, but protect the quality of the environment, agricultural species as well as wildlife species. And if there's any way that we can incorporate them into our risk assessment, I think that would be beneficial. For this talk today, I'm going to focus mainly on anthropogenic chemicals, which we call toxicants or pollutants that are present in the water. And I will not be looking at some of the other types of contamination that we may find. So we'll be focusing on emerging pollutants, things like pharmaceuticals, plastics, herbicides, pesticides, etc. And really the area where we have kind of a blind spot are with certain chemicals that have just been introduced into our environment, or we have limited data. And these we group into contaminants of emerging concern. And so these are chemicals that we know are present in water, in food, in soil, in air. So we know we are being exposed. We just don't know what the level of concern should be. So these contaminants of emerging concern can come from food production, such as agricultural chemicals, packaging materials, things like pesticides and plasticizers, the BPA, some of the perfluoroalkyl substances. These could come from industrial activity, things like metals, polycyclic aromatic compounds. We can also start to find, depending on the water source, we can find pharmaceuticals in the environment. Things like drugs, as well as things that are added into different formulations, plasticizers, etc., and personal care products. Things like DEET, phthalates, again, the perfluoroalkyl substances. All of these are being found in different types of water samples. And what we're concerned with are those compounds that, yes, are present, but also those that tend to persist in the environment. They don't biodegrade. And also, because of that, and because of some of the physical properties of these types of chemicals, they tend to bioaccumulate in wildlife species, and they may be present in food that we consume. Another concern is that we are all exposed to complex mixtures. So we are exposed to all of these chemicals that are shown on this list here, plus degradation products and metabolites that are present from all of these chemicals. And so we have to start thinking about the interaction between the chemicals and not just each individual molecule by itself. And really, for a lot of these chemicals, we know they're present, but we do not know what the impact is on our health or on the health of the environment. So how do we start to tackle the presence of these chemicals in our environment, and how this can be addressed, and how we can communicate the issues? So the traditional approach is shown on this slide. And this is basically just taking your sample through either grab sampling or passive sampling. And then you have to go through a standard set of parameters that are measured in these samples. And some of the samples may require extraction and concentration methods in order to be able to detect. But the key feature is that we have a certain list of these parameters that we are mandated to look at. And we examine the concentration of these chemicals in our water. And then we have certain drinking water standards that are regulated, that are telling us what decisions need to be made with that water. So in the US, the main standards are set by the Safe Drinking Water Act as it pertains to, again, drinking water. And they have dictated maximum contaminant levels, or MCLs, for a variety of different chemicals. And I'll show you a list of some of these in the next slide. So through this Safe Drinking Water Act, the EPA has set standards for states and local authorities, water suppliers, et cetera, for the levels that are acceptable of a variety of different pollutants and different nutrients, minerals, metals, et cetera. So when we're talking about some of these contaminants of emerging concern, and talking about the Safe Drinking Water Act, we really have limited information on certain of these chemicals. So right now in the Safe Drinking Water Act, we have maximum contaminant levels for about 90 different contaminants. And these would include disinfectants, disinfectant byproducts, organic chemicals, microorganisms, organic chemicals, and radionucleotides. So if you look at this table, this is just kind of an excerpt of this multi-page document. It'll show you the chemical and what would be a maximum contaminant level for that particular molecule. And it'll also tell you what the potential risks are if you exceed that concentration over an extended period of time. So again, this is a relatively limited list of chemicals. There is kind of a waiting list for contaminants, and this is called the candidate list. And this too goes through iterations. So they have proposed 66 chemicals in this candidate list, and they're asking for more information so that they can move it into this regulated list of contaminants. But as of right now, these chemicals do not have an MCL set. But if you do have an MCL for a chemical and you have measured that contaminant in water, if the sample is greater than, the concentration is greater than the MCL, then a warning or an action is required. If it's less than this, it's simply just deemed acceptable. So these are very simple to follow guidance. They're revised with new information. So some of these MCLs will go down over time or be altered in some way. And again, some chemicals will be added. Some problems with this, again, many chemicals that we know we are exposed to, there is no MCL, and we do not have sufficient data to to tell us if there is a risk if we do detect it. Also, many compounds are present in low levels in the environment. So for some of these chemicals, we have to start thinking about our analytical techniques, and can we actually detect the chemicals at these low levels? And then finally, a very important concept is that we are exposed to thousands of chemicals in our water, and add this into what we're exposed to in our diet and in our general environment. So we have to start thinking about interactions between chemicals that are present. And that is not addressed by this particular approach. So this is where we have to start thinking about mechanism-based bioassays. How do some of these chemicals cause the potential health effects that are being observed that are driving these regulatory features? So let's start talking about mechanism-based bioassays. And the framework we're going to use is the framework that the EPA and other agencies are using to look at how chemicals are able to lead to an adverse event. And so these are just a concept of linked events that occur within a cell, within an organism, that leads to some outcome that we do not want to see that was initiated by a chemical or a contaminant. So this process starts with the chemical entering the organism and interacting with some feature of that organism, usually some macromolecule in a cell, that is initiating a chain of events. And so this molecular initiating event may be an interaction between that contaminant and a receptor, or interacting with DNA, or interacting with an enzyme. That interaction is interpreted by the cell in a variety of different ways, depending on what the molecular initiating event is. So some of the key events that you might see are changes in gene expression, altered tissue function, altered cell proliferation. Again, these are causally linked to the presence of that chemical and that chemical interacting with this molecular initiating event. These key events then are tied to organ effects and organism effects that over time result in the adverse event within that organism. Some disease affecting development, affecting population size if you're looking at wildlife species. So this is a very easy framework that we can go through. And what we're trying to do is characterize the key events that are necessary in order for the disease to occur. And we can use this for some key adverse outcomes and some key molecular initiating events to try to link exposure to a chemical with an adverse outcome. So when we're talking about adverse outcome pathways that are important for environmental pollutants and some of these contaminants that are found in water, there are several that we can think about. We can think about the effects on DNA damage leading to cancer. We can think about effects on cardiac function. There's just a variety of different endpoints that we can be concerned with. The overriding concern that we hear from the public and from the regulators is this idea of endocrine disruption. And I like to think of this as really a bigger type of adverse outcome. We have endocrine and metabolic disruption that is all part of one kind of signature pathway. This adverse outcome has multiple types of toxic responses that we want to avoid. Things like reproductive effects, proliferation and cell proliferation and cancer, interactions between different molecules that are affecting half-life of chemicals and hormones in the body, metabolic disorders, things like diabetes and obesity, that could all be linked to xenobiotics. And these xenobiotics, again, we can consider these endocrine disruptors or metabolic disruptors, or think of them holistically. But when we talk about a chemical that is an endocrine disruptor, we are saying that the molecular initiating event is an interaction between that chemical and a receptor for hormones within that organism. And so there are several that we are concerned with. Androgen receptor, which binds with testosterone, estrogen receptor, which of course binds to estrogen, thyroid hormone. There are a variety of these different endocrine-related molecular initiating events. And how these lead to a toxic response is by changing enzyme activity, transcription factor activity, and metabolism that together are altering steroid levels and how the cell responds to steroids, leading to things like reproductive deficits, and leads to the potential to increase risk of developing cancer. Also linked to this are a series of receptors that have really evolved to respond to foreign molecules. And how our body responds to foreign molecules is we recognize a foreign chemical structure, and then we try to induce the enzymes that break down that chemical so that we can excrete it. Well, some of these chemicals are going to be regulating enzymes that are going to disrupt steroid levels, and they're also going to alter fat metabolism and foreign molecule metabolism, leading to their own set of toxic responses. So long story short, we can cover a lot of different potentially hazardous chemicals just by knowing whether they interact with some of these key molecular initiating events. And again, these are a variety of different types of molecules. We tend to focus on nuclear receptors. That is our forte. And again, very important for this adverse outcome pathway. And I'll be going through some of these assays in a little bit. Okay, so if we know a chemical is interacting with some of these molecular initiating events, then we would predict that these are going to lead to some adverse event within that organism. Well, how can you screen whether a compound, either singly or as a mixture, is regulating some of these molecular initiating events? Well, one of the approaches that we take is using mechanism-based bioassays. And these are really focusing on molecular initiating events, but you're creating an assay system that can respond to that chemical and that interaction with the receptor and drive its key event regulating gene expression in a way that's very easy to measure. So what we do is we develop cell systems that express the molecular initiating event, as well as what's called a reporter gene, which will be able to interrogate whether the receptor has interacted with a particular chemical molecule that has a sufficient structure. So what we do is we're building these artificial cell lines that will tell you yes or no, we're getting exposed to a particular chemical. And then under the right conditions, you can also answer questions about how strong of an activator of the receptor is it. And I won't go into any detail, but we can build these for a variety of different molecular initiating events. We can make them very specific. We can make these for different species. We can make them even for mutants. So we have a lot of flexibility to how we develop these very sensitive and robust types of assays. Okay. And we'll come back to some of this in a little bit. So these mechanism-based assays, so we have a series of these adverse outcome pathways. And now we want to start looking at chemicals that are present in the environment, and are they interacting with these particular molecular initiating events that are predictive of adverse outcomes. And there are two ways that we'll talk about. One way is simply using the data that's already in the database that tells you that this chemical does interact with this particular molecular initiating event and what concentration is required to lead to a biological response. And we'll call this the EAR response. There is data out there and ways of interrogating that data to look at individual chemicals as well as mixtures. But as we'll see, there's some limitations to using publicly available data because you are at the mercy of the quality of that data and the quality of how you interact with that, what your search terms are and what information you're getting from it. We tend to rely more on cell-based bioassays. where we can look at, yes, individual chemicals. We can get a lot of information, but we can also look at complex mixtures. So some things that we're going to be talking about is using these cell-based assays, but certainly you may have a lot of questions about, well, if I want to go into using cell-based bioassays to look at water quality, how do I pick a source of those bioassays? Some individuals who have been working for this for a long time already have kind of their own homemade assays, but you may not have that. And so you have to start hitting the commercial resources and start to ask some specific questions, such as what is the sensitivity? How low of a concentration can you go with these assays and be able to detect a signal? Reproducibility from lab to lab, from person to person, from lot to lot. How much of a response do you see? The robustness. How available are these? Can you easily just call up and order different assays? How many assays are available from a retailer? Is it just human? Is it other species, et cetera? Cost, ease of use, how much technical detail do you need? And then you also want to start thinking about which assays you choose. You are definitely going to want to start with those molecular initiating events that are known to interact with compounds present in the environment and are predictive of some adverse outcome, like endocrine disruption. You're going to want to know that these assays have been used for environmental samples before, and you have all of the technology that's required to perform these assays in your lab. Technology, expertise, just overall resources. And then, of course, if you have assays that are standardized with reference compounds, have the reference compounds available, if you have information about trigger values, all of these things are going to go into your decision-making process. But let me just tell you a little bit about kind of the nuts and bolts for how this is done. Again, this is part of the overall workflow that we're going to go through. You collect your samples, you extract them and concentrate them. And now you are asking the question, is there something present in this sample that's able to regulate this molecular initiating event that's associated with toxicity? So in these cell-based bioassays, it's relatively simple. You pick what molecular initiating event, what receptor you're interested in. And there are some relatively straightforward biochemical and cell biology techniques. You thaw the cells, you plate them into a 96-well plate like what is shown over here. You put them in an incubator. So you have to have some sterile technique associated with this. And then you treat these cells that are present in each well of the plate with your test compound. This could be dilutions of your mixture. These could be a variety of different sites or time points, et cetera. And you put these back into the incubator. While it's in the incubator, your chemical is going to be interacting with the molecular initiating event and driving the expression of luciferase. After you've let it incubate for a sufficient amount of time, then all you're really doing is you're adding the detection reagent for the reporter gene and reading the amount of the reporter gene. So the amount of luciferase activity that you get corresponds to the amount of activation of the receptor, and that's proportional to the activity of the chemical that you added to it. So what we have shown up here is some typical single chemical concentrations for chemicals that are known to activate the androgen receptor. And you can also look for antagonism if you have a constant amount of an agonist and then add in another chemical during this incubation step. So very simple to interpret. You can get a lot of data from each of these dose response curves. When we are doing environmental monitoring, generally speaking, what we are doing is we have a series of wells that we set aside to look at the reference dose response curve. And then what we are going to be doing is comparing the amount of luciferase activity we get from our environmental samples relative to this dose response curve. And this is something called the bioequivalent quotient. And again, this is something we'll talk about in more detail. So again, relatively simple assays, very powerful, very robust, a lot of different possibilities for different molecular initiating events that you can look at. But again, you have to look at your expertise, how much you have in your lab, what resources, and what you need to look for a retailer to get these into your laboratory. Okay, so now we've talked a little bit about just kind of the framework, what the problem are, what some of the approaches are, keeping in mind mechanisms for how chemicals are able to lead to an adverse event. So let's start making some decisions. Let's start looking at water. Let's focus on those chemicals of emerging concern that we don't have a lot of information. And let's start with what is probably the easiest for most people that work in this area that are already doing chemical analysis, but want to start incorporating in mixture effects as well as adding in some of these adverse outcome pathway decision making. So again, what we're trying to do is use mechanisms. So for the next part of my talk, we're actually going to go through and talk about 3 different ways that we can approach making decisions about water quality. So we're going to assume at this point that we have our samples collected, we have them extracted, and we've already sent these out for our normal analytical chemistry. So we may have a list of chemicals that we know are present, that we're concerned with, that we have been working with before as far as an analytical lab, know all of their different techniques, and they're following all the appropriate guidelines. And so what we're trying to do is things like prioritize sites for cleanups, thinking about chemicals and bioactivities that might be a concern, focusing on adverse outcome pathways, and at the end making a decision about water quality. We'll start with the exposure activity ratios, or EARs. This is using simply the concentration of the chemical that's present and looking at a publicly available database. Then we'll go into the bioassays and looking at cumulative effects of an extract in the cell-based assays and incorporating this information into the adverse outcome pathway. And finally, taking these bioassays and looking at what's called a trigger value and using bioequivalence. Okay, so let's start with the EAR approach. And again, this is probably a good entry for many people to start looking at mechanisms, because it's utilizing data that's already been curated and put into the EPA CompTox dashboard. This was previously called the ToxCast dashboard. And so what we're going to be doing is comparing the concentration that we have of a particular chemical in the water, and this could be in micromolar or it could be in micrograms per liter. And you're comparing this to a measure of potency of that chemical for a particular molecular initiating event. And this is called an AC50, so activity concentration, where you're getting a 50% maximum response. And so this EAR is simply a ratio of the concentration relative to the potency. The higher the EAR means that you have a higher concentration relative to the effect level. So you have more concern the higher the ratio, the exposure activity ratio. And that's shown down here. So we have a compound that is highly exposed but low bioactivity. We wouldn't be concerned with that. A compound with low exposure but high bioactivity, again, we don't worry about it. We're not exposed to it. But as we start going to compounds that are present at higher exposure, and we have a high level of bioactivity, where we start to get concerned is where we have overlap between the concentrations required for bioactivity relative to what's present in our water samples. So the higher priority, higher the EAR value. So again, we are simply taking the concentration that we have in the water, we're interrogating the database, and we're saying for all of the known effects of X chemical, what is this EAR for molecular initiating event 1, molecular initiating event 2, etc. So what we are looking at is actually the summation of these ERs. So we could take all of the different ER ratios that are present for a chemical. Let's say it's interacting with 3 different molecular initiating events. We sum those together to get a way to rank chemicals. The higher the ER summation, the more risk it would be. We can also start to look at pathways. How many chemicals that are present in our sample interact with the same molecular initiating event? Estrogen receptor. How many ERs do we have for chemicals interacting with the estrogen receptor? So we can start to rank pathways. We can add up all the ERs for all the chemicals at a particular site and compare sites. So we have a way of kind of ranking different aspects of our water sample using concentration and effect. So here's an example that we've done the sort of ER approach. These are from surface water samples from collaborators here at Penn State. These are surface water samples that are would be impacted by private septic systems as well as agricultural activities. So we have a list of compounds that we're interested in, including personal care products and pesticides. We've actually gone out and measured the concentration and then looked at the ToxCast database. And I'll get back to this, but not all the chemicals that we've detected have information that is publicly available. So here's a graph that you might commonly see in people that are doing simply chemical analysis at a site. So these are showing the concentration of the chemicals in micrograms per liter, actually across, we looked at 9 different sites at 8 different times here. So we're kind of looking at the high and the low of each chemical across the 10 sites. So we know the concentration. If you have an MTL for chemicals that are in your list, at this point you can look at it and say, is there a MTL level for these chemicals? And so I just highlighted one of these chemicals, the agricultural product atrazine. And we know that the MTL for this is 3 micrograms per liter. You can look at this and see that we are below the MTL for this particular compound, but we don't have MTLs for many of these others. Now, if we use the ER approach and look at each chemical and sum up the ERs for all the pathways they interact with, you start to see differences in how you prioritize chemicals. Because now we're seeing chemicals that may not be present at high levels, but they have a high level of biological activity. And so compounds like I've highlighted here may not be things that would jump out at you for being a concern from a pure microgram per liter basis, but you can see that they are an issue relative to their biological potency as predicted by the public database. But also notice that we have a blind spot. We have chemicals that we know are present at multiple sites, but we have no biological information in order to get an idea about potential risk. So again, some very useful aspects here. We can start to prioritize, the chemical concentration, the chemical and the ER values across different sites that are shown here. We can start to prioritize sites that would be of concern. And we can also start to incorporate our mechanism-based assays as well. So if you remember back to the adverse outcome pathway and the molecular initiating events, we had a series of receptors that are important for endocrine disruption. And we're seeing here that we have several of these genes, the androgen receptor, estrogen receptor, and in this case, this is the glucocorticoid receptor, are predicted to be increased activity based on chemical concentration alone and their ability to interact with these receptors. So concern from an endocrine disruption, and also concern about some key events that occur that might alter endocrine metabolism as well as lead to metabolic disruption. So some concerns here, we can start to prioritize which pathways are of most concern. But you can see that we're now taking advantage of known mechanism for these chemicals, and we can start to prioritize sites and times and chemicals based on their ability to interact with these receptors. So what are some of the features of this EAR approach? Well, many chemicals that we looked at were present above the limit of detection in these waters, so they were there. We can look at time, site-dependent differences in chemical concentration. We unfortunately cannot get EAR values, even though we know the chemicals are present. We cannot get EAR values because the CompTox database does not have sufficient data in order for us to predict. So again, we can start to use this to prioritize what types of chemicals, potential sources for where they're coming from, start to look at what chemicals are of concern, et cetera. But it's really, again, the fact that this is theoretical, that there are these black holes that are present there, causes some concern. I think it does give some information. So let's see how we can add some sophistication to this and start using bioassays. So this is really just focusing on what I showed before for how reporter assays work. And in this case, we're just focusing on how chemicals are interacting with one molecular initiating event and how we actually quantify this in a way that we can start incorporating it into decision making. So what I've shown here is one single molecular initiating event, a nuclear receptor. And we have 3 chemicals that are known to be ligands or regulators of this particular nuclear receptor. So we can look at each of these chemicals alone and see that they lead to a dose response curve. Or what we can do is look at a mixture that these chemicals are present and get an idea of the activity within that sample that is the result of the mixture. And just to give you an idea, these 3 chemicals by themselves, each one of them has a different dose response characteristic. So this first chemical may have a high level of activity but is present at a low dose. This chemical is medium as far as potency, but is present at a higher concentration. And this chemical has low efficacy, but it's present at a high concentration. And what we're looking at is we're developing a quantitation for what is the contribution of each of these 3 chemicals relative to this one receptor. And this is what's called a bioequivalent quotient. So we are looking at the level of activity relative to a reference compound for that molecular initiating event. So if this were the androgen receptor that we were looking at, we have a reference compound, this dihydrotestosterone, where we have a known dose response. We know what the EC50 value is for this. We can model this curve. And now if we take our sample and we're getting 200 luciferase units, we can say that that is equivalent to that cell having 12.6 nanograms per liter of DHT. So these are called bioequivalents. And the effect that we would see on the cell from this mixture would be the same as what we would see from 12.6 nanograms per liter of dihydrotestosterone, because we're looking at the mixture of all of these bioequivalents from these chemicals. So how can we use these bioassays? So here's a couple of approaches that have been used. And again, we're just looking at comparing luciferase activity, comparing bioequivalent quotients between different conditions. What's showing up in the upper left-hand corner here is a situation where we have wastewater treatment water before and after we go through some remediation. In this case, it was manganese oxide from this paper that's shown down here. And we can see that we have a decrease in bioequivalence, a decrease in androgen receptor activity after we've remediated. We can use this bioequivalent approach to look at different water samples. In this case, these are road sprays that are being applied to decrease dust from different rural roads. And we can start to prioritize sources of these water, of these aqueous solution, based on which ones have the highest levels of bioequivalent for aryl hydrocarbon receptor activity, something that we don't want to have. So some of them are non-detectable and some are equivalent to X amount of dioxin per sample. want to look a little bit over on the right-hand side here. So this is that study set that I showed you from the beginning that we did the ear analysis on. And so the ear analysis, when we looked at the potential pathways, it predicted that there could be an increase in the androgen receptor activity. So what we did here is we actually looked at all of these different molecular initiating events. And these are involved in things like endocrine disruption, xenobiotic metabolism, lipid metabolism, basal metabolism, toxicity, and then we have zebrafish assays as well. So we can look to see, are we getting activity in these samples for these important molecular initiating events? The chemical analysis predicted AR, and we did see an increase in activity of AR. And we can see this in both humans as well as in zebrafish. So this is something that may be a concern for environmental species. But we have picked up activity that was completely unpredicted from the ear analysis. Mineral corticoid receptor activity. This is a receptor that's involved in blood pressure and basal metabolism. And we can see here that we have significant activity here. Okay, so we've opened up things. Same way that we use the ear, we can now look at effects on different pathways like endocrine disruption, metabolic pathways, simply by adding up the BEQs for each of the different receptors. So here is an example of endocrine disruption, all of the different androgen receptors by site. We can also look at these same things in zebrafish. So we can start to compare species, compare responses, prioritize chemicals, prioritize sites. But this is non-theoretical. This is actually being measured within these particular samples and not dependent on someone else doing the research. So these bioassays give you a lot of information that can be used for looking at different time points, different samples, different remediation efforts. They're telling you changes in bioactivity that are associated with important endpoints. These are very sensitive. We can go below the necessary level for many compounds that do have the MTLs already determined. And importantly, these are cumulative effects. So we are looking at multiple chemicals that are acting through the same pathway that together would lead to a concern instead of looking at each individual. Not dependent on other people's data. This is real versus virtual. We can rank and prioritize, and this improves with your ability to communicate about safety. You could say things like there's less estrogenic activity, less potential for endocrine disruption after we've remediated, or there is more concern about X site than there is with this other site. So you can make some decisions, but sometimes it's difficult to say how much of an effect is enough. Just the fact that you can detect activity, at what level do you start to actually make decisions and take action? And that's where the effect-based trigger values come in. And again, this is how much of an effect is the concern. Do we want to go by limited detection, which is commonly the background level of activity plus 3 standard deviations? Do we go by the level of quantitation, such as the background plus 10 standard deviation? Well, this is really just technical. It's not really saying anything about the level of biological effect. And that's where effect-based trigger values are becoming more and more important, and is really becoming the focus of how we're using these the mechanism-based assays. And this is looking at the potential to cause risk using approaches that are very common for anyone doing risk assessment. So you are simply setting a reference dose. And if you're looking at drinking water, you're looking at the reference dose and then putting in factors like acceptable daily intake and trying to set, well, what concentration is required given a certain amount of body weight and extrapolation data, just like how you do with normal risk assessment. What is different in this approach is that we are using the mechanism to set what the reference dose is. So for example, if you are concerned about endocrine disruption, you are setting the reference dose to be the reference compound for that molecular initiating event. So if you are concerned with the endocrine disruption through estrogen receptor, you're going to use estradiol, that reference dose, to determine the drinking water trigger effect. Then you're going to use the BEQs from your water to say whether you have exceeded that reference dose. And as with any type of trigger value, this is constantly going to be reevaluated as we know more about what would be considered an acceptable dose. And it's very similar if you're talking about surface water, but in this case, we're using more guidance values and mixture effects based on point of departure from some of the molecular initiating events. And I'll go over this in a little more detail. So for some of the endpoints, we have enough information where we can set an effects-based trigger value for compounds that known toxic effect for the reference compound. And we can actually set effects-based trigger levels for either human consumption as well as ecological effects. Again, based on the amount of information that we have on the reference compound relative to the molecular initiating event. So we have a variety of different effects-based trigger levels for some of the bioassays that are currently available that we can use to set a guideline. If we do not have an EBT, then what is commonly used is an effective concentration 10%. For those of you who have worked in risk assessment, this is a common point of departure dose in a dose response curve. And it is appropriate if there is no set EBT. And so again, what we have this trigger dose relative to the reference compound, and we are looking at our sample's BEQ in terms of this reference compound to determine whether we've exceeded the value or not. If our BEQ in our sample is less than the trigger value, then no action is required. And of course, if the BEQ is greater than this trigger level, then further action is required once you go through the confirmation. Okay. So here's a couple of examples of how this can be used. So in the first case, this is that hospital wastewater example that I showed you before for two different molecular initiating events. One is the androgen receptor and the other is the estrogen receptor. And what I've shown you is the effect-based trigger level. And then we have two different sources of the wastewater, HM and CH, before treatment and after treatment, before treatment and after treatment. And you can see here that with the androgen receptor, this really doesn't require a lot of our attention because even in the absence of remediation, it's below that trigger level. But this is much different than what we see with estrogen activity. So we're far above that trigger level in the untreated wastewater. So what we have to now evaluate is whether our remediation, our manganese oxide oxidation step, is able to bring that trigger value down. And so obviously we need to focus on estrogenic compounds and the ability to get below that trigger value. We can use this in environmental samples as well. Might have different trigger values depending on what endpoint you're looking at. This is estrogen receptor beta, and I've used the EC10 value for this. And you can see here if you're looking at a time point, so these are different times of collection, you can see here something happened at this particular time that led to an increase in estrogenic activity. So you may want to go back and take a look at what happened then. Was there some spraying activity that occurred if this is a stream near an agricultural site? Was there some accident that occurred, et cetera? So you have something that exceeded the EBT that may have caused some environmental damage. Here is a case where we've looked at several sites and looking at different molecular initiating events. This particular site has crossed over on the EBT value, again, for the estrogen receptor. So what is going on at this site? And you can also start to look at multiple factors and how close we are to the EBT for multiple different endpoints as well. Okay, so this effect-based trigger level is an important new approach. We can use it for drinking water, various types of environmental water as well. We may just have to adjust what the concentration is. If there is no EBT yet, we can use the point of departure dose of an EC10. This is assuming, of course, that there is a threshold of toxicity. You probably wouldn't use this for DNA damage, for example. The other thing to think about with this approach too is why did a particular sample cross over the EBT value? And many times that's because you have the presence of a very potent molecule within that sample. So estrogen is a great example of that. We find 17-beta estradiol in a lot of wastewater treatment facilities. We find things like estrone within agricultural water as well. So there's some causative agents that we know would be present that would be triggering concern. The other thing that, at least in my view, helps with communication if you do have EBTs is you can point out the fact that, yes, you can detect it, but it's not above a level that is of concern. So, you often are able to detect certain chemicals and just by the fact that you can measure it drives up people's concern that they're at risk. So these are help with communication and kind of decreasing the fear that's often associated with detection. And again, we can give clear guidance. If the BEQ is greater than the EBT, then further action is necessary for that particular site or that particular time. And this is really becoming the acceptable direction in how we are using effect-based monitoring to look at water quality, because it is adding functional data as well as quantitative data into our decisions. All right, to conclude, so effect-based monitoring, what we've been talking about, either EARS or more frequently these bioassays, is something that's increasing in popularity, although it's really only being used right now from a regulatory standpoint in the state of California, where they're using ER and AHR to examine recycled water quality and whether it can be used for recycling. But this is expanding, and more states are moving this way. In the EU, there's also a lot of push to use effect-based biomonitoring to assess water quality. But certainly these give you a lot of good information about potential pathways, potential chemicals of concern. A lot of prioritization can be done for water quality at different stages of purification, et cetera. So, even though there aren't regulations in a lot of places, it's growing, and you can also get a lot of information, and you can also allay consumers' concerns about what is present and what should be of concern and what should not. But of course, and I mentioned this at the beginning, there are a lot of bioassays that are available. You really have to be careful which ones you choose. Choose those that are definitely representative of water samples, so they're commonly detected. They're associated with an adverse outcome. They are available in the species you're interested in. They're readily available for the technology you have available. As we start looking at the individual EBTs, there's also some interest in trying to look at multiple EBTs in order to get a better understanding of water quality. So instead of just looking at endocrine disruption, start including other types of toxicity as well, so that we're not just limiting it to one toxic endpoint. We have to be concerned with all potential toxic effects within the water. And we need more research. We need to come up with more trigger values. We need to understand which pathways are of the most concern, which molecular initiating events are representative of the widest variety of different types of water samples. So with that, I'd like to thank you for your attention. We will have time for questions and answers. And if you can, please write questions into the chat box or contact us later. Thank you.
All right, excellent. Thanks so much, Jack, for a great presentation. All right, so we'll jump right in here. First question, Jack. So if you could only perform 2 or 3 assays for water quality, what do you think they would be?
As I said in the presentation, I think you have to kind of look at the assays based on whether they represent a adverse outcome pathway that you are concerned with. It has to be something that is readily available and you have the technology to examine it in your lab. And it would have to have some trigger value so that you actually can make some decisions. So at least with the state of California, how they've approached those issues is they've kind of focused in on the aryl hydrocarbon receptor, AHR, as well as the estrogen receptor, as the two that they are using to implement these bioassays. If I were to expand it a little bit, I would probably add the androgen receptor into that short list because of the importance in endocrine disruption, the sensitivity and the concern, and maybe PXR, just because it represents a receptor that responds to a wide variety of different foreign molecules.
Excellent, great point. All right, another really nice question here. What specialized equipment and training would you need to run these bioassays?
So there is some cell culture that is required, but luckily a lot of the commercially available kits require just short-term cell culture. So you would need a laminar flow hood or a sterile hood. You'd need the capability to pipette reproducibly into a 96 well plate and a luminometer or a plate reader of some sort to read the reporter gene. So there is some biochemical techniques that you'd need and some specialized equipment, but usually those are things that you'll find in a lot of cell biology labs, life science type labs.
Excellent. And I think in the interest of time, we'll just have one last question here, maybe one looking towards the future, but what are some of the developments in cell-based assays that you're seeing?
Well, I think part of it has to do with how you approach using the cell-based assays. And I mentioned this at the end of the talk, having trigger levels that are helping you make decisions. So that's been a big push, is trying to figure out what are the trigger levels for some of these types of pathways that we know are being affected. The other thing is to try to find assays that are responsive to different adverse outcome pathways. So we've so far just kind of focused on the endocrine and metabolic disruption, but we also have to start looking at other types of toxicity that are seen with pesticides, for example, with neurological effects or DNA damage types of assays for looking for carcinogenic compounds.
Excellent. Well, great answer. Jack, thanks so much for all your insights today. It's really been a pleasure speaking with you.
Thank you.
Excellent. Yeah, my pleasure. Obviously, a really big thanks also to the audience for joining us today. And last but not least, we'd also thank Indigo Biosciences for sponsoring the event. And in closing, we hope you enjoyed this scientist.com and Indigo Biosciences webinar, and we'll see you again next time. Have a great day, everyone.